Okay, I'm going to do a quick time check. So it's going to freak out on you for a second. How long have I been recording? 11 minutes? We're good. Okay, going to continue into the endomembrane system. So, the endomembrane system is a eukaryotic feature. It is the membrane-bound organelles inside of eukaryotic cells. They are the nucleus, the endoplasmic reticulum, which is my favorite one to say, the Golgi apparatus, the lysosomes, vesicles and vacuoles, and then the plasma membrane. Obviously, everything has plasma membranes, but it's a little more extensive in eukaryotic cells. So, nucleus first. Nucleus holds your DNA. DNA is organized into chromosomes. Humans have 46. We have 23 pairs, 23 from your mom, 23 from your dad. Other animals have more, less. Doesn't really matter in terms of complexity. But each chromosome is going to have a DNA strand coiled around proteins. The proteins help to keep it, they're called histone proteins. They help to keep the chromatin, the DNA, condensed, so it fits in the nucleus when it's not needed. The DNA will kind of unwrap from those proteins when it's being expressed and made into proteins. When we have all of this together, we call it chromatin. You're going to see chromatin, chromosomes, even chromatid later. In general, when you see them, think DNA. The difference between a chromosome and a chromatid we'll talk about more extensively in Unit 5. Nucleus also has a double membrane, so it actually has two lipid bilayers on it that we call the nuclear envelope. It's just the protection for the nucleus. It has a bunch of holes in it that are the nuclear pores that allow stuff to enter and leave. So it's where your RNA would leave when it is done. It's where the proteins needed for DNA replication or RNA synthesis would enter and exit. Additionally, we have the nucleolus, which is essentially just a really DNA-rich region where the ribosomes are synthesized. Ribosomes are hugely important. You can't make other proteins until you've made your ribosomes, so got to make those first. And that is done in the nucleolus. Endoplasmic reticulum. This is kind of the post office of the cell. It is essentially just a bunch of membranes with specific proteins embedded in it. It merges directly with the nucleus, so things can exit the nucleus straight into the endoplasmic reticulum. And the whole purpose of this huge folding structure is to increase surface area. Is it this unit that we talk about, surface area? I think it is. It is. Surface area will be a huge theme of this unit. A lot of the things our cells do are to increase their surface area. So we get all these folds, we get a bunch of extra surface area. Extra surface area means we get a bunch of extra proteins that we can fit. And it makes all of our reactions happen efficiently and quickly because everything's kind of localized in one small area. We split the ER, endoplasmic reticulum, into two regions, smooth versus rough. The only difference is if it has ribosomes or not. The rough ER is a protein synthesis area. That's why it has ribosomes. The ribosomes will build the polypeptide, and then the polypeptide will be inside of the endoplasmic reticulum, inside what we call the lumen, so just the in-between space of the ER. And then I called it the kind of post office. No, this one's the factory. This one's more of a factory. So we make the proteins, we manufacture the proteins, and then we have to kind of ship them off to whatever stores are going to sell our product, which is the protein in this case. So the endoplasmic reticulum makes the product, it makes the protein, and then it ships it out. It might need to do some modifications to keep it safe and intact on its way to its destination, or maybe put a marker on it so that we know what it is. But it's kind of just assembling and sending it off to the next place. Usually, there are 3 destinations for the proteins here. One, they can be secreted, just put out into the bloodstream, into the extracellular space. You can think like in our stomach to digest food. They can be incorporated into the cell membrane, so transport proteins, receptor proteins. And additionally, they can enter lysosomes, which we'll talk about in a bit, and help to break stuff down, do some cellular digestion. However, while the rough ER has a ton of ribosomes on it, it is not all of our ribosomes. There are plenty that are just in the cytoplasm. Generally, though, if they're made by ribosomes in the cytoplasm, they tend to be used in the cytoplasm. We don't have much control over where they go if they're made in the cytoplasm, because there's no structure around it. Smooth ER, if you'll notice, a little further away from the nucleus. That doesn't really matter, but this is a big lipid synthesis area. So fats, steroids, phospholipids. Sometimes it'll do some detoxification, but generally think about it as lipid synthesis. The rough ER is the more exciting one, but in both cases, we're making some macromolecules. Okay, the Golgi apparatus. This one is the post office. So after leaving the ER, the endoplasmic reticulum, not the emergency room, many transport vesicles go to the Golgi apparatus, sometimes called the Golgi body. And this is where we modify proteins, we store proteins, and then we send them off in other transport vesicles. It is also made up of a bunch of membranes that kind of form these channels and little pockets called cisternae. You do not need to stress about the cis side versus the trans side, but generally understand that the Golgi body will receive proteins or vesicles, whatever is in them, could be lipids, from the ER. It will undergo modifications. The Golgi body will do whatever it needs to do to it, and then it ships it out on the other side, pinching off, forming new vesicles to transport elsewhere in the cell. So it doesn't make proteins, but it modifies and kind of regulates where they go. Helps us specialize, helps us compartmentalize. Most of the time, if we're sending something to Golgi, it is because it is going to be either taken out of the cell, exported from the cell, secreted by the cell, or put into the cell membrane, so it has a specific destination it wants to get to. Sometimes, though, they will just become lysosomes as well. A lysosome, now we finally can talk about it, is a vesicle, so just a membrane-bound sphere, with hydrolytic enzymes, so enzymes that do hydrolysis, that break down macromolecules, usually not in plants. We usually make them in the rough ER, modify them in the Golgi. The Golgi then makes the lysosomes. And a key feature of these is that they are acidic inside the lysosome. They're kind of like a little cell stomach in a lot of ways. And so their enzymes are acidic as well. There's a lot of different reasons why we have lysosomes. They do a lot of things. They will fuse with food vacuoles and break down food, help to break things down from bigger macromolecules into smaller ones that our cell can use for respiration or building blocks for other materials. But they also help to break down old cell components, other organelles that might be damaged, not working as well, so that everything can be reused. Kind of a big recycling center. Anytime we have molecules that need to kind of be reset, destroyed, turned into base nutrients, that's the lysosome. Additionally, oh no, my image. They have an important role in apoptosis, which is cell death, programmed cell death. Essentially, a cell has some sort of defect and self-destructs. And in this case, the lysosomes just release their enzymes, and their enzymes just start to break down everything in the cell. They don't know that it's a cell. They just break down everything around them. Vacuoles are similar to vesicles and lysosomes. They are just large vesicles, large membrane-bound spheroid structures. They have a few functions. One, food. They hold food and other material that we want. And in certain organisms, we'll have contractile vacuole, which is in order to keep an excess of water out. So it kind of, instead of the cell bursting, the contractile vacuole will swell and burst instead, and the cell can go on living. Plants have a special vacuole called the central vacuole. In each cell, it stores water, nutrients, pigment, waste, huge storage area. We do not have them in animal cells. So this is your big summary of the endomembrane system. Essentially, you can kind of divide it into consumption versus secretion. So secretion starts at the rough ER. We make the proteins, package them in vesicles, send them to the Golgi apparatus. The Golgi apparatus makes some modifications, and then it sends them to their destination, being a lysosome or an enzyme, a lysosome or a secretory vesicle where it will get released into the blood. If it makes a lysosome, that meets up with our consumption pathway, breaks down food. And remember, the ER also helps to synthesize lipids. Ultimately, why do we care about this? The endomembrane system really is what enabled eukaryotic cells to become more complex and multicellular life to even emerge. The main thing is our cells are not, as far as we know, conscious. They can't actively, mindfully control the reactions that happen inside them. The chemical reactions happen based on whatever is around them. And so if you have all these membranes and internal compartments, you're minimizing the amount of time where you're doing chemical reactions that are not helpful to you. It wouldn't be helpful to do cellular respiration completely in the cytoplasm, where half the time you're not breaking down glucose into energy, you're doing something else. So reducing that and then just increasing surface area for reactions, giving more space for the reactions to happen that you want to happen, is hugely important. Makes the cells much, much, much more efficient. So that's kind of the big idea, is this compartmentalization of these cells, of the eukaryotic cells, allowed them to specialize, become more efficient, and more complex. Here's your interactive questions. This one's fun. We have 6 feet of DNA in each cell. How the heck does it fit into that small space? Think about it. Go through. I believe I'm going to stop this one here. Yeah, definitely stop this one here. We'll hit the next 2, which are going to be the energy organelles, and then all the rest of the organelles in the next video. Went a little rambly towards the end, but I'm going to hit under 25 minutes. So, as always, that's all, folks. I'll see you next time. Check out my cool hat.